Okay, good afternoon, everyone. Uh, thank you. Thank you, the organizers, for giving me the opportunity to speak in this symposium. It's a great honor for me. So to just begin with, I am today going to speak on the proteome-wide analysis of mammary epithelial cells, reveals protein signatures associated with lactogenic differentiation. Before I begin my talk, let me speak about where we are and where I am. Basically, I am from Indian Veterinary Research Institute. This is a university and also Institute of Importance in Veterinary and Animal Science Research. And our main campus is in Ijat Nagar, and we have the oldest campus in India in veterinary research layer, where it is Mukteswar. And it is located in northern part of India, if I say. This is in the state of Uttarakhand. So the great Himalayan, this is the great Himalayan range where we are located, and you can see these Himalayan peaks. And this was established in the year 1893. So it's more than 130 years old. So this is the mother campus, which was subsequently shifted to its main campus. And the work that I'm going to present actually has been carried out in National Dairy Research Institute, Karnal. Again, it is located nearer to Delhi. That is another university. I have just shifted recently. So this center is famous for its virology research and temperate animal husbandry research. Great legendary Dr. Robert Koch, way back before 1890, he visited this place just to train the then scientist about microbiology. So with this background, I'm just going to start about this problem. What is the genesis of the problem? So as we understand, the bovine mammary gland is constituted of network of ducts, which ultimately ends up in alveoli, and the innermost layer of the alveolar epithelial is constituted by alveolar epithelial cells. So these alveolar epithelial cells are mammary epithelial cells. They reflect the milk-producing ability of the farm animals. And especially in case of when the animal is giving milk, most of the epithelial cells are terminally differentiated. So these terminally differentiated MEC are involved in synthesis and secretion of milk proteins. So the background is when the animal is giving milk, it is going through a specific lactation curve, like early lactation, where it reaches the peak and subsequently the milk yield is persistent and it goes down. When this kind of phenomena is happening, that means there are many proteins and genes and various molecules which are being expressed inside the system. When you talk of milk yield and relate to genetics, we see that this is the protein has ultimately, the gene has ultimately to be expressed, but it has to be expressed in a system. So because the epithelial cells are the precursor cells for the synthesis and secretion of milk, we understand that majority of the milk-related genes and proteins are expressed in mammary epithelial cells. So during this lactation curve, expression of protein at specific time points, and ultimately when the animal is at its peak of lactation, we understand that many of the genes are expressed at optimal level. So we also understand that means this will be reflected in terms of proteins. And ultimately those proteins have to be maintained to keep the process of lactogenesis going, so that this will constitute the lactation persistency. So in one way, milk producing ability differs among different animals. How to show, especially in the Indian context, we see that high producing cattle are mostly Western breeds, but the indigenous breeds like Bos indicus, they have a difference in the milk yield to the extent of 5-10 times. Lowest and highest milk producers. In this case, what happens? We expect So the genesis of problem, I thought probably we can find out some of the proteins which are getting expressed in the mammary epithelial cells during lactation, and which may give some idea, can you find out some biomarkers which are associated with milk yield and lactation persistency. So what we did is we started our journey by isolating the epithelial cells from mammary epithelial cells from milk. So epithelials from milk, then subjected them to high separation two-dimensional jelly electrophoresis and liquid chromatography, identification of protein by mass spec. Then we went for deep proteome, global proteome. What are the number of proteins expressed at a specific time points, and differential proteome. Say, for example, different stages of lactation, high and versus low producing cattle. So the main purpose was to identify probable biomarkers associated with lactation function, maybe peak yield and lactation persistency. So we followed the technique like took milk, somatic cells, epithelial cells almost constitute 5% of the total cells. And we use anti-cytokeratin antibody, which is specific. I mean, cytokeratin 18-8 are specific to the cell surface of the epithelial cells, which are captured by use of anti-cytokeratin antibody. And the secondary antibody was coated on the surface of magnetic beads. So this way, captured many epithelial cells and subsequently saw that you can see the magnetic beads are sitting over the epithelial cells and check that purity. So purity of the epithelial cells were checked under microscope as well as gene expression assay by using various milk-specific genes, alpha-lactal proteins, kappa-casein, beta-lacglobulin, epithelial membrane antigen, and went for checking whether actually the enrichment of the epithelial cells has taken place. So subsequently went for Western blot just to check again the cells are fine. So we just wanted to see in a physiological lactation what are the various proteins which are expressed in the mammary epithelial cells. We understand that if a cell is growing, many proteins will be expressed. Either transcript level, we expect many genes to be expressed, but at the protein level, the final end product will be relatively less. So when you grow a actively proliferating cells versus differentiated cell, we expect that in a physiological lactation, only selected proteins should be expressed. So as per our expectation, this was fine. So this was subsequently published in Proteomics. So we followed a conventional strategy, two-dimensional jelly electrophoresis by following standard protocol and prepared a reference map of the mammary epithelial cells which are isolated from milk. In the beginning, even if I identified 900 different spots were picked up, hardly you could identify 158 proteins. Subsequently, we went for subcellular fractionation, where cytosolic membrane nuclear cytoskeletal fractions were taken, and the proteins were extracted, subjected to SDS phase in gel digestion, and subjected to mass spectrometry, LC, ESI, MS, MS. This is another shotgun proteomics by cutting the proteins from the gel, and these are the various subcellular fractions protein in SDS phase. So when we identified almost 508 proteins from the 300 proteins, the total was 600 proteins. So the main purpose of saying is this: we may expect many proteins in a proliferating cells. I'll come this afterwards. But in a physiological lactation, when I isolate the epithelials from milk, which are terminally differentiated, the number of proteins are likely to be less, which is here. And subsequently, we subjected them to gene ontology pathway and network analysis, identified some pathways, 45 pathways. And just to know so many proteins are there, we have to find some way out to understand their role during this process. So many of the proteins which positively regulate lactation were identified. which maintains the cell shape and which helps in the cell survival because they are required. And cell difference is required during the differentiation of the milk production will take place. And negative regulation of apoptosis will help the cell to again survive. Likewise, we went for a network analysis by Pathway Studio. So this was just a small study just to understand whether are we able to identify some proteins in the physiological lactation which are associated with either milk yield or performing some function in cells which are coming in the milk. And subsequently, our main interest, can we find out some proteins which are differentially regulated in high versus low producing cattle or during different stages of lactation. So this was published in PLOS One. Again, here we followed, again we isolated the epithelial cells. These are dynabeads, you can see, and cells are sitting over the, dynabeads are sitting over the mammary epithelial cells. And we took knowingly 3 different groups, like a low producer, high yielder, this is Bos indicus, sahiwal breed. Current breed is a crossbred cattle in India, National Dairy Research Institute, where I worked before 5 months. So they are supposed to be high producers. And we isolated the epithelial cells, extracted the protein, and subsequently prepared the samples. By making various combinations, we subjected them to in-gel differential analysis. This is fluorescent-based differential expression of proteins, where you can see in a co-detection manner whether a protein is getting highly expressed or lowly expressed. And so then they're subjected to mass spectrometry, and we could identify some proteins which are actually differentially expressed both in high and low producing cattle. And they maintain a sequence, like say, these are the proteins. We selected one, milk, fat, global epithelial growth factor 8, lactoadherine proteins, which got expressed highly in the high producing cattle. If I go back, if we can see low producing, even if it is high producer, it is in between the crossbred and the low producing cattle. So we just try to see whether a proper ratio is being maintained, and very nicely they're making the maintained. And subsequently, those proteins actually help us to decide further strategy study actually how the differentiation regulates the milk production. So we subjected the principal component analysis where low producer, high producer animals, they were made distinct groups, and interestingly, low producing cattle, we can find many of the proteins are upregulated. That means they may have some kind of inhibitory effect. We went for some kind of validation by RT-PCR, by isolating mRNA and subjected them to RT-PCR, qPCR, and we saw that is really expressed very highly. If you see this, MFG8 is highly expressed to the level of 47-fold change. And here you can see some proteins which are actually highly expressed in low producing cattle. So just to sum up this study, although there are so many, serpin-like proteins are highly expressed in low producing cattle, MFG8, high expression high yield animals as compared to low yield. So actually, and there are many other proteins which are also expressed, differentially expressed, and we thought, so these molecules can subsequently be validated. So in order to understand the function of these kind of differentially expressed proteins, a cell line is required. Although there are cell lines from bovine and other species, we thought of generating our own cell line, because buffalo milk constitute almost 57%, 55% of the total milk production in India, and globally also this is a major milk producing breed. So we established the buffalo mammary epithelial cells for the first time, which was published in PLOS One. typical dome-shaped epithelial cells along with various having cobblestone morphology and various domes you can see, which were indicative of the purity and of the epithelial cells. And subsequently, they're characterized where you can see the fluorescent image of cytokeratin 18, and some vimentin expression was also there, and we use some negative control also. Subsequently, this cell line we are using for further studies. So what we did is, this cell line we use for generating what are the various proteins, which like here in case in-depth proteome analysis of more than 12,500 proteins in buffalo mammary epithelial cells 9 identifies protein signature for active proliferation and lactation. So as I told, when we are isolating the epithelial cells from milk, we did identify hardly 600 proteins. But here, we took the actively proliferating buffalo epithelial cells, mammary epithelial cells, and by deep proteome analysis could identify more than 12,500 proteins. So subsequently, what was done is, we took the epithelial cells, then went for subcellular fractionations like cytosolic, membranous organelle, nuclear fractions, and cytoskeletal. Even if we went for secretome analysis, the proteins were trypsin digested and peptides were fractionated and subsequently subjected to mass spectrometry and bioinformatics analysis was done. So if you see this one, we did identify 12,609 non-redundant proteins. Cytosolic 8,000, membranous proteins almost 6,000, nuclear 5,000. Likewise, there are many overlap in fact, and we went for a Venn diagram where the overlapping area represents protein share among respective fractions. At total number 85 proteins are common among all the fractions. Just to make the long story short, the cake analysis suggested that these proteins were associated with 325 molecular pathways. Some of the highly enriched molecular pathways observed were metabolic pathways, MAPK, PI3AKT, insulin, estrogen, signaling pathways, and we identified some newly proteins in this study which are reported to be involved in Notch signaling, transport, and secretion processes. And interestingly, significant number of 372 of uncharacterized proteins are found in buffalo mammary epithelial proteome database, were found in the BUMEC proteome database, which are uncharacterized. So from here, we thought of, because when we subject the epithelial cells for proliferation, the galactogenic differentiation process is not that much activated. So we try to mimic the lactation conditions. So here we went for TMT-based differential proteome analysis to understand the molecular basis of functional differentiation of buffalo mammary epithelial cells. So here this is many different which are published recently in PASEB. And here we followed basically three different approaches. This one is, this diagram depicts the experimental plan to standardize the prolactin concentration for optimal differentiation of BUMEC, because prolactin is essential for the lactogenic differentiation. Different concentration of prolactin was taken, and one specific concentration identified to be when the beta-casein expression was optimally expressed. Subsequently, this diagram depicts the sample preparation procedure for differential proteome analysis of proliferating and differentiated BUMEC cells using TMT labeling. So I'll be coming to this. Different days were taken, and here the differential proteome analysis of proliferating and differentiated BUMECs using TMT labeling And the identified proteins were subsequently validated. Only selected protein 11 differentially expressed proteins were validated by qPCR. You see this proliferating Boumex treated with lactogenic hormones like 3, 6, 12, and 15 days. For 15 days, and mammary epithelial cells derived from buffalo milk was subjected for qPCR. So if you see this one, to study the effect of different concentration prolactin on expression level of casein 2 in Boumex, because beta casein is an indicator of differentiation. So here, if you see that this is the prolactin concentration and the transcript level of beta casein. When it was used at 8 microgram per ml, the expression of beta casein was very high. And this is further validated by Western blot using the beta casein, and you can see how the concentration is rising over a period of time. So here the qPCR analysis showed that various concentrations was used, and 8 microgram concentration was used. That led to optimal differentiation. If you see the differentiation of Boumex cultured on collagen 4-coated plate in the presence of lactogenic hormones, this is typical buffalo mammary epithelial cells, where you can see the cobblestone morphology of the cells. So this is a phase contrast image of proliferating cells grown in normal growth up to 70% colorpoint stage. And this is phase contrast grown in the presence of lactogenic hormone. You can see 8 microgram per ml bovine insulin hydrocortisone in collagen 4-coated substrate. So red arrows depict the domes, you can see, indicative of the lactogenic differentiation in epithelial cells. Subsequently, you can see how the domes were formed, morphological changes in Boumex during differentiation at different time points. If you see control 3rd, 6th, 9th, 12th, and 15th day, numerous days treated, the expression number of domes went on rising and subsequently decreased by 15th day. So this gave an indication. We selected few days just to see what are the various proteins which are changing when the lactogenic differentiation is going on in vitro. So this mimics rather the lactogenic condition, although the system influence is not there because it is grown in a cultured cells. So subsequently, we went for identification of proteins, and we identified, we identify almost this one slide is missing. We identified 4,900 differentially expressed proteins. So we took various different time points, like control 3rd, 6th, 9th, 12th, and 15th. I think we went for 3rd, 6th, 12th, and 15th day, just to check how the protein expression is behaving. And here, the control, the 6th day, 12th day, 3rd day, how the shape of the domes are changing. So once we identify the proteins, then we prepared a scatter plot for this. This is a PC analysis for differentially expressed point 3rd, 6th, 12th, 15th day versus control in differentiated set population, which shows the proteins are actually getting differentially expressed, but they're very much scattered. And this shows how the heterogeneous nature. This 2D plot shows top 2 components of PCA with most significantly determined proteins, and but they are highly scattered, which depicts their heterogeneous in nature. The top 20 highly differentially expressed proteins are subsequently was subjected for a scatter component of PCA analysis with rank plot distribution mapping, and it shows how the protein expression changes sequentially during different days of lactogenic differentiation. We prepared a hierarchical cluster analysis of the expression profile of the proteins, And you can clearly see each row in the heat map represents a protein, and each column represents the fold chains at different days of treatment, 3, 6, 12, and 15. The up and down regulated proteins are indicated in red and blue colors. So on 12th day, you can see majority of the proteins are upregulated. And as the days goes from 3 to 15, there is a continuous change in the expression of number of various proteins. Here, the best 4 clusters, we clustered them because proteins are so much, almost 5,000 proteins. It is virtually impossible to take all the proteins. So we just made different clusters based on simulating the expression pattern of differential expression separately, expanded and shown with reported number of proteins involved, where you can see in cluster 1, 72 proteins, in cluster 2, 34, cluster 3, 49, and cluster 4, 67. And their expression pattern also you can see how they're changing, but they're making a kind of sequence. Subsequently, these proteins were clustered. Network analysis of isolated 4 cluster in hierarchical analysis cluster. There is cluster 1, cluster 2, cluster 3, cluster 4 like days 3, days 6. In between 6 to 9, and here this is 15th day. So those proteins are clustered there. They were subsequently went for protein-protein interaction study. The network was made. If you see the correlation analysis of differential proteins and metabolites using Medscape, using different differentiation of Boomex. In fact, number of proteins are less. We just use various bioinformatics tools to make some sense. So complex network comprise input proteins, spherical nodes you can see, and dark blues, metabolites, hexagons, and reactions. So kind of reactome you can see, which are making edges and nodes in order to depict something. And then went for identification analysis of clusters and modules from protein-protein interaction network using Cytoscape plugin, MCode, where you can see many of the proteins. We understood that ribosomal proteins are getting highly expressed, RNA splicing, mRNA processing proteins. And in module 3, so module 1, module 2, and module 3, endocytic proteins you can see, and they're making good interaction with each other, and proteasomal proteins. So when the cell are either either cell is growing or cell is differentiating, many of these proteins making interaction, and that is a biological process. Having taken all those kind of proteins, then we validated 11 differentially expressed protein using qPCR. And you can see them that the expression of 11 differential expression studied at mRNA level among proliferating Boomex like control, again 3, 6, 12, and 15 days. And MEC, not only cell culture cells, we have taken also cells which are isolated from milk of buffaloes, because this is a buffalo mammary cell line. So MEC derived from buffalo milk using qPCR. And the expression level of genes are normalized. So here we can see that whatever observation we have made in the cultured cells, and we could clearly observe the expression of those few selected proteins in the epithelial cells which are isolated from buffalo milk, suggesting that the differentiation process has a definite role with the milk production. So many of the cellular events were affected during differentiation of buffalo mammary epithelial cells, which are involved in ultrastructural reorganization and functional differentiation, and some of which are responsible for cell polarity, maybe cell polarity factors like tight junction, apical end, basal ends, and these are some of the various pathways we did observe. So out of so many proteins, after deep analysis, we did find that probably we can list some selected proteins about which nothing much is known their role about differentiation, especially lactogenic difference. So we can see some of the, we noted down some of these proteins, around 10, 11 proteins, and thought of validating one. So overall, out of these 5,000 proteins, out of which 600 proteins were differentially expressed, and various informatics analysis, we made schematic illustration of proposed major molecular events during differentiation, and the differentially expressed proteins identified in the are represented in red colored spherical nodes. And this is just kind of hypothesis proposing that some kind of mechanism is going on inside the epithelial cells during lactogenic differentiation in vitro, which may also be true during physiological lactation. Then subsequently, we thought of, okay, let us see whether can we find something like involucrin. Here, this is the control cells. And in the differentiated cells, involucrin is very highly expressed. And here, we used an inhibitor. We used an inhibitor retinoic acid. When added to the cultured cells, expression was reduced. And we see the expression of beta-casein, how it is rising. So that means it has some role during differentiation. One more thing is, it has been recently published that in the differentiated cells, this is multinucleated, and the aurora kinase is responsible for this. We try to see the differentiated cells which are generated are having any multinucleated cells. Not many we did find, but there are few epithelial cells we can find multinucleated epithelial cells. And low number of multinucleated epithelial cells can be attributed because here the system doesn't work. Here only we are growing in vitro cultured cells. That's why the number of differentiated, multinucleated cells are less. This can be one explanation. So subsequently, because as I understood, in the epithelial cells which are isolated from milk of cattle, we did find out MFGE8 also. And we thought definitely it has some role because its role is already known, but we try to understand exactly what exactly it is doing inside the cell during lactation. So although it may not be directly related in the milk production, but it plays a major role in signaling in order to influence the milk yield and to some extent lactation persistency, as well as maintaining the mammary health. So the other term is lactadherin, MFGE8, milk stimulated macrophages localization, plasma concentration also, and mode of action, the physiologic function, as a breeze ligand, the lectin-like domains engage phosphatidyl L-serine, the second EGF domain engage this one, so leading to phagocytosis of apoptotic cells. Many of the apoptotic, till now it is reported in this clears the apoptotic cells. But here in high producing cattle we found out, so it may have some relevance. Probably many of these cells may be apoptotic and it may be getting clear. So it is a 52 kilodalton protein. It plays a significant role during apoptosis of mammary epithelials during involution, and hence in remodeling of the mammary gland. You see the mammary adipose tissue in puberty, when it goes, ductules are formed, and gestation, the alveolar epithelial cells are moved. During parturition, it is there, and during involution. It has been seen that during involution, MFG8 is more, although it is not directly related to apoptosis, but it sometimes clears the apoptotic cells. But it was not very clear how come in a lactating animal, the MFG8 expression is more, especially there is a difference in high versus low producing cattle. And interestingly, when we went for differential expression analysis in a lactogenically differentiated epithelial cells, the expression of MFG8 was also more. So this made us very curious to know what may be the possible function. So what we did, we made some hypotheses, like this regulates cell cycle growth and apoptosis in mammary epithelial cells. If you see this one, the MFG8 is getting secreted. It may have effect as well as molecular process unknown. And during early involution, this comes out, and this may be somehow doing some kind of activity, and pathway was not known. So our approach was to go for cell culture model, buffalo mammary epithelial cells, and go for a stable shRNA transfection, mass spectrometry, functional activity relationship, various bioinformatics software was used, and did some cellular assays like BRD, MTT, neutral lead caspase. So SH-based gene silencing image, we all know, went for this one, where we can find when the cell was subjected to shRNA-based gene silencing, we can see the changes. Changes in the sense, there is down regulation of MABG8 in the shRNA silenced buffalo mammary epithelial cells, which has a fold change, 86-fold down regulated. And we can see they are the control cells, perfect morphology. These are the scrambled. So the vector in which the MFG8 shRNA was cloned, this did not contain the shRNA, only the vector was there. So perfect morphology, and we can see the GFP. But in which there was shRNA, we can see the fluorescence was less. And subsequently, as the days went by, you can see this is the control. These are the control. And if you see this one, transfected epithelial cells with MFG8, specific shRNA containing this one, 24 hours. This is a 24 hours picture. These are the control lines. And this is a 72 hours picture. We can see some kind of domes. And in 120 hours, you can see the cells are losing its morphology. Some kind of change is taking place, and there is aggregation of these cells, epithelial cells. So when there is silencing of the MFG8 gene, some kind of cellular architecture is getting altered, thereby making kind of aggregation. So we wanted to know what are the various proteins which are getting expressed in these kind of cells when the shRNA-based silencing of the epithelial cells has taken place. So we designed this strategy like significant important proteins we identified, knockdown of MFG8 to determine the cellular function, and subsequently went for validation. So this experimental design quantitative proteomic workflow allows a complete analysis of updated proteome. If you see this one, these are the knockdown cells here. You can see these are control cells, and qPCR we did, and various assays like phagocytotic assay, stable transfection, then in vitro phagocytic assay, cell growth confluence assay, wound healing assay, and transwell migration assay. Many different cellular assay was performed, and we checked their possible functions. Majorly, as I told, this is involved in a kind of signaling. So we more focused because we identified almost somewhere around, again, 12,000 proteins, but we identified the differentially expressed proteins you can see are many, almost 4,000-5,000, and we try to find out what are the various transcription factors. So all the identified significant differentially expressed protein one-fold change up and down regulated in 938 proteins for the identification of associated transcription factors involved in cell cycle regulation. And this Venn diagram was prepared, and subsequently comparison of knockdown and control cells was done by MTU, BRDU, caspase, and Western blot analysis. And from these kind of studies, we could understand that this protein is not involved in apoptosis. Rather, it is involved in kind of signaling pathway. As we know that AKT-PI3 kinase pathway, STAT3 and STAT5 pathways are quite prevalent, and especially in case of ruminants, the lactation process is gone by the mTOR pathways. So it is somehow when the MFG8 is being silenced because of alteration in the signaling pathways, there is perturbation of the signaling pathway, and there is altered expression of various proteins. That means this may be major molecule, because we have also seen the expression of when there is knockdown of the MFGE8. We saw that almost 95% of the beta casein is not expressed. That means it has a definite role. So apart from its role in the mammary gland remodeling, it may have a definite role in the lactation process as well. So, and then we made kind of a postulated systemic pathway for MFG8 and downstream regulators, and try to find out. So just to sum up my entire story, I'll say that initially began with epithelial cells isolated from milk, tried to see whether in the physiologically mammary epithelial cells can I find out some proteins or no. Went for differential expression analysis, selected few proteins, then check whether they, for that we generated mammary epithelial cells, and we are using this as a cell model to understand the functions of various proteins. And in actively proliferating epithelial cells, for the first time, we identified largest number of proteins ever reported in a single cell, not even in human and any other cell line, that constituted more than 12,500 proteins, and identified differentially expressed protein in lactogenically differentiated epithelial cells, and subsequently validated them by using some new proteins like involucrin and MFG8, which is already known, but we wanted to see whether it has any role in the process of lactation and kind of various signaling pathways. So, so just to sum up, MFG8 suppression results in reduced growth of mammary epithelial cells by autocrine or paracrine mechanism, results in induction of transcription factors such as cyclin D1, FOXA1, ZNF652, aurora kinase B, and many others for modulating the cytoskeleton-associated proteins and cell cycle regulators. And it plays a role in signaling mammary gland developmental remodeling. And this study, through proteomic examination, has underscored its importance in cellular homeostasis. So this study will not be possible without the support of my students. They're all PhD scholars. He's in USA. He's a faculty. Vijay Salin is a postdoc. Sweta is a postdoc in USA. Azmal is a PhD scholar, recently joined as EMBL fellow. Manoj again is a faculty and is a PhD student, and he is a young faculty working with me. And this work will not be possible without the support of Department of Biotechnology, Government of India, Department of Science and Technology, National Agricultural Innovation Project, and National Agricultural Science Fund, and in fact NDRI, because I worked here for more than 30 years. And thank you so much for giving me the opportunity for presenting my talk. And questions, please. Have any question?